This is the Chapter 10, Segment 4 outline for behavioral neuroscience. We're going to get started with color vision. So it's been known that birds, fishes, and insects have extraordinary color vision, and as well as not quite as good, but pretty close, humans and old world monkeys have pretty good color vision as well. This is because they all have trichromatic vision, which involves 3 classes of cone pigments. However, most mammals actually have very poor vision because they have dichromatic color vision, which involves only 2 classes of cone pigments. These mammals can distinguish only between a short wavelength, which is blue, from a long wavelength, which is not blue. We can also bring up that colorblind humans also have dichromatic vision. Colorblindness is actually more common in men because men only have 1 X chromosome, whereas women have 2 X chromosomes, and the gene for colorblindness only is on the X chromosome, and since men can only have 1 X chromosome, they don't have a backup X chromosome to compensate. Recording from retinal ganglion cells of old monkeys shows second stage processing of color vision. And now we're going to get into the process of vision. Ganglion cells and parvocellular layers of LGN cells fire in response to wavelength. However, can also be inhibited by other wavelengths, and this is a Herring prediction. Neurons in LGN cells respond to wavelengths with at least 600 nanometers in length because L is sensitive. It can be inhibited. They are inhibited at shorter wavelengths. There's an equation where it's plus L divided by minus M cell. An example of a spectrally opponent cell in which 2 regions of spectrum have opposite effect on the rate a neuron fires in action potential. Bipolar cells give input to ganglion cells. They each have 2 to 3 different cones. 1 cone is excitatory, and those from the other type are inhibitory. Spectrally opponent ganglion cells record the difference in stimulation of different population cones. Peaked M and L are not different. However, plus M over negative L and positive L over positive M cells yield distinctly different neural response curves. Most important role of complex vision is the ability to denote which parts of complex image belong to one object versus another. Especially opponent cortical cells provide 3rd stage of color vision system, and this is done by adding and subtracting outputs of spectrally opponent ganglion cells. These yield red versus green, blue versus yellow, and black versus white. Spectral responses on cell correspond to wavelength of principal hues. Some retinal ganglion cells respond to certain direction of motion of objects. They may only respond if objects move left but not right. That's just an example. Some monkeys and humans in the cortex in V5, which is also known as the medial temporal area, respond to moving visual stimuli. It's also been shown that experimental lesions in V5 impaired the performance temporarily. However, all stimulation of V5 doesn't improve performance. It can actually result in seeing objects move in different directions than the direction that the object is moving in real life. There's a hypothesis that there are 2 visual systems. One system involves the identification of objects, which is located in the visual cortex. And the second system involves the location of objects, which is controlled by the superior colliculus. 2 researchers, Mishkin and Ungerhed, proposed primates have 2 main cortical processing streams originating in the primary visual cortex. The first one is that ventral processing stream for visually identifying objects, and the second is the dorsal stream for guiding movement and placing location of object. They believe that there were many cross connections between these 2 streams. All right, now we're going to get into possible many disorders and diseases of the eye. First one is a really rare one. It's called optic axia. And in this disorder, patients have difficulty using their vision to reach and grasp for objects. And this results from damage to the posterior parietal cortex. The next one, and then also in this, we're going to bring up that the anterior part of the visual stream merges with the motor cortex and includes visual and motor neurons, and are activated by not only doing an action but also watching someone else perform an action. These are known as mirror neurons. All right, our next disorder is myopia, which is known as nearsightedness. This results from when the eyeball is too long, and therefore the eyes now need to focus on objects that are closer to them instead of farther away because distant objects appear blurry. The eye has to focus on these objects in front of the retina instead of on the retina due to the enlargement of the eye. They believe that certain environmental factors cause the eyes to grow in excess, and one of the main theories of environmental factors causing excess eye growth is indoor lighting, because indoor lighting does not match the composition of wavelengths that outdoor lighting does. And this is probably why a lot of people with myopia can't stand outdoor lighting. Amblyopia is a misalignment of the two eyes, otherwise known as lazy eye. The eye and the retina are normal, but acuity is poor. Basically, the one eye is going to kind of look off into another direction, or maybe possibly near the nose. If not corrected in very early childhood, the primary visual cortex will suppress the info that enters the cortex from the eye. Basically, it's kind of like adaptation, because if that eye is constantly looking off in a different direction, it could mess up, kind of going back to the conflicting sensory theory. The brain will purposefully shut off that info from the eye to kind of keep that from the brain becoming confused. So therefore the eye would become blind. They believe that balance of eye muscles can be surgically adjusted to align both eyes. Another option is by training and covering the good eye to restore vision to the lazy eye. Basically, it's putting an eye patch on the good eye, which will force the bad eye to kind of refocus to where it's supposed to be. However, this method has been kind of controversial because it's been proven that just using normal glasses is more effective than using the eye patches. Our final disorder is macular degeneration, which is the loss of central vision due to damage of photoreceptors in the retina due to atrophy in the retinal pigmented epithelium. One way to go about correcting macular degeneration is to transplant new photoreceptors, although it's kind of risky because they could be rejected. Another option is to culture cells that aren't photoreceptors, inject them into the eye, in which they can actually become new photoreceptors inside the eye. A third option is to use a virus to induce retinal bipolar cells to express light-sensitive ion channels, in which case these light-sensitive ion channels will replace photoreceptors.